cannot be presented by the X-ray crystal structures, but by AFM only. So this is based on your observations only, right? Or is it
Well,
the truth?
yeah, yeah. I mean, it is not okay. So, X-ray crystallography couldn't identify a second bind for serotonin transporter for a long time. Yeah, although there was always the suspicion that there is, and they succeeded 2016. Yeah, and we showed before with AFM that there's two binding sites. Yeah? We also showed with AFM that there is two binding sites in the dopamine transporter, but for the dopamine transporter so far they couldn't prove it with X-ray crystallography that there's two binding sites, although there's also clear suspicions. And our speculation is that why was it not possible to show it with the dopamine transporter? Because the binding forces to the vestibular binding site, to the ST2 site, are so weak, and are weaker for the serotonin transporter. This is of course speculation. But
Because not getting a crystal, there could be other reasons as well.
no, they are getting the crystal. They are getting the crystal. They are getting the crystal. But they don't see binding to the S2 side. I mean, I can show you. I show the crystal. They are getting the crystal. That's not the problem. It's not that they don't succeed. Here you see the crystals. Yeah. And this is now, but with dopamine and this is cocaine. This is a crystal with cocaine. Yeah. But cocaine is only in the central binding site. Yeah. They're not, or here dopamine. Here is the crystal structure. They are all published in Nature. Yeah. Here's the crystal structure with dopamine, but also only in the central binding site. Yeah. They're getting the crystal. But, and for the third, okay, now we have this problem. I can try to switch. And for the serotonin transporter, I just want to show this picture. And in the meantime, for the serotonin transporter, they see it here in the central, so with escitalopram, this is the escitalopram, so exactly the molecule we used, and this shows the binding to the central binding site, and this shows binding to the vestibular binding site. So with CERT they get the crystal with the two binding sites, but that only with one.
Thank you.
Yeah, so this was nanopharmacology, if you want, like this.
Attaching drugs. So in general, this strategy of putting drug molecules on the peg linker, you say it's a bit more challenging than putting proteins in
Well, the challenging part is to synthesize a drug that is biochemically active. Yeah, so for us it's not more difficult because we couple it through AIM. So what we are using either click chemistry, and what we also using is amines actually also bind to a Malay linker at certain conditions, and this is also what these two binding strategies. No, for us it's not more difficult. Just for AIM it's very difficult to synthesize it. As long as someone can synthesize it, yeah. And also the art is to synthesize it in a way that they are still binding, because these are all very small drugs, and if you modify it, it can of course influence the binding, and this is.
In general, from pharmacological perspective, is it a good strategy for screening drugs? Because that
No, no, I don't think, no, because it's way too time consuming. So single molecule for spectroscopy and screening doesn't fit together.
what can be interesting is verifying sensors.
Exactly, yeah. Exactly, yeah. To really get the energy landscape, for instance, something like this is of course interesting. And also to discover these weak transient binding sites, that is, for that, this is difficult with other assays, but for that it is very good.
Peter, one more question. So for such kind of experiments, what's the concentration of protein that you use on a glass surface?
On the glass surface?
Yes.
No, we are measuring on live cells. We're expressing serotonin and dopamine transporter.
Okay.
And it's a. I mean, we didn't really measure the surface concentration. So that is our setting. So we express. I mean, AFM is very difficult to do on neurons because they are, you don't have a clear surface. This is where they are, and so this is what the guys in Vienna are doing. They are expressing certain DUT on CHO cells, and CHO cells somehow turned out to be. We really like them for AFM. We use it also in the next talk. I will show. We were also using CHO cells because they don't form tethers. This is what happens with some cells. If you punch into them, a tether forms, but this is not the case with the CHO cells. If the expression level is not too high, we recently also had a problem when the expression level was too high, also with some tether formation, but that is not the case here. And these are very clean experiments actually, because it has it has basically no unspecific adhesion. If I think. Did I show that?
Status can run into microns. Really long.
The length can be long, yeah. Usually it is something like 300 nanometers, yeah. But this is just deformation. I mean, we also have to be aware if we do AFM on a cell, yeah. We compress them by it could be 1 micrometer that we compress them, because the spring constant of the cell is much lower than for a cantilever. Yeah.
So whenever you start with such experiments, for example, either with protein or cells, so do you go for hit-and trials, that how much or how much the concentration should be, or you have a defined number for it?
What we are doing, I mean, but I have it in another talk. This is all, yeah, and I should say also the mutations are done by the guys in Vienna. So what we have is all the transporter have a GFP tag. Yeah? So what Rong does, he looks at the cell in the fluorescence microscope, and then he chooses a cell with a high expression level, and this is where he does the experiments. And then we always have sufficient binding. So this correlates. Yeah? So the binding probability correlates with the expression level, which we can detect with the fluorescence microscope.
Thank you.
Huh?
So the force resolution needed to distinguish between two binding sites, does it depend only on the thermal noise in your measurement, or are there any other factors?
No, it's not. And this is, do we have that in the, did I put that? So the force resolution is around 2 piconewton. Yeah. So because in this, you can somehow, let me go to, you can somehow, it is somehow similar as with fluorescence microscopy. Yeah. So, okay. So we can distinguish here 12 piconewton, 14 piconewton. I didn't put in the standard deviation here, but it's around 2 piconewton. And what is similar with fluorescence, because it's quite interesting. So these are kind of Gaussian function, all these peaks, and this is also what you get from a Slingler fluorophore. Yeah, you get a Gaussian function, and the width of the Gaussian function is something like, like wavelength numerical aperture or whatever, is something like 250 nanometers. Yeah? And this is the resolution. But if you want to depict the position, it's only dependent on how accurately you can fit the Gaussian function. And this is the positional accuracy. Okay? And that is kind of similar here, because you see this widespread of data, yeah, which is because of the stochastic nature of our binding. Yeah? But to depict the most probable force of unbinding is also only just how accurately you can fit the Gaussian curve. Yeah? And the standard deviation is then sigma, the width of the Gaussian curve, divided by the square root of n, and n is the number of experiments. And this brings it down to 2 piconewton. Yeah? And so this is why we have a force resolution of about 2 piconewton here. Yeah? And that is now here very important.
Thermal noise could be little higher than 2 piconewtons.
Yeah. Exactly, in the end, yeah. But it comes out to about, but if we would do 10 times more experiments, then also the the SD should go down in a way, yeah, because then you kind of average. But that is the same with fluorescence microscopy. Exactly. Although it's a completely different event, but the statistical description is very similar. Okay. Welcome back. We are switching now from pharmacology to cancer defense. Of course, it's not cancer as general, it goes more into immune therapy. And the title is Nanomechanical Binding Mechanism of Ligands Drives Agonistic Activity. And this is the work of Hannah. And she actually just gave me the slides of her PhD defense. So what you hear now, so more than 1 year ago, so what you hear now is basically, so I'm now, I'm candidate and I have to defend the PhD thesis with your questions. Okay, good. So I'm talking about CD40 and CD40 ligand receptor pair, and also some therapeutic antibodies. And actually the data evaluation here was quite intricate, and I think it's also very interesting from this aspect. But first, let's go a little bit into biology. So CD40 is a member of the tumor necrosis factor receptor family, and it occurs on antigen-presenting cells, which are macrophages, dendritic cells, B cells, but also on epithelial cells, platelets, and tumor cells. So it has a lot of functions. And the CD40 ligand, also called CD154, is the physiological ligand of CD40. And it's mainly expressed on activated T lymphocytes. And from the structure, it is a stable homotrimer and has 3 possible binding sites with CD40. Yeah, so a little bit similar as the spike protein, which also is also a trimer. But interestingly, only 2 of the 3 possible binding sites of CD40L are occupied by CD40. That has been found in X-ray crystallography. Yeah? So, of course, you would expect all 3 sites, but for whatever reason, they couldn't identify some conformational change there in the X-ray structures, but for some reason, only 2 of them can bind. So, and then, of course, binding of CD40L to CD40 induces something, which is a clustering of CD40 in the cell membrane. Yeah? And then a really complicated cascade starts. I just go on it superficially. So there is a downstream signaling in the antigen-presenting cells. There is also a TRIF adapter proteins, and which is recruited that leads then to this clustering. And then it induces upregulation of major histocompatibility complex. It leads to increased expression of co-stimulatory molecules that are important for enhanced antigen presentation, which are these ones. And then it induces the release of T-stimulatory cytokines that are important for T-cell activation. Yeah? And this is how it helps with cancer, because basically it upregulates the immune system. Okay? So that is the function. But then there is also therapeutic antibodies, which are supposed to induce the same as CD40 ligands, basically also induce the clustering, but of course antibodies are only bivalent molecules. Yeah. Okay. Yeah, these therapeutic antibodies, you know the structure. Antibodies consist of 2 FAB arms and one Fc arm, and the two FEP arms can bind to the antigen. And this is what the antibodies are supposed to do. They are supposed to lead to clustering. Yeah. If they bind to two CD40s and somehow induce the clustering. But antibodies, so IgGs, so human IgGs, but there's subclasses of antibodies. Okay, so IgG1, 4, 2A, and 2B. There's more, but this is the 4 subclasses we tackled. And these subclasses of antibodies, they have different flexibilities here in the hinge region. Yeah? And this depends on the number of interhinged disulfide bonds and disulfide bridges between FAP arms and hinge region. Yeah? And the flexibility of these 4 subclasses is like this. So IgG1 has the highest, then IgG4, no, sorry, the other way around. IgG2b have the highest flexibility, and sorry, I'm confused. IgG2b is the most rigid one, yeah, and IgG1 is the most flexible one. Okay. And interestingly, they show different agonistic activity.
These are structural flexibility.
These are structural, yeah. So this is not related to these therapeutic antibodies. This is related generally to the subclasses of the IgGs. So the paratope can be different, yeah. But it is just, you see it indicated here because of the structure here in the hinge regions, yeah, the disulfide bonds. And there's an inverse correlation between agonism and flexibility. So this means the stiffest, the least flexible antibody has the highest agonistic activity. So this has been found. Okay. And there's also FC epsilon receptor cross-linking might lead to additional stabilization of the IgGs. And there's also other studies that found that somehow the affinity, there was actually a Nature paper last year, they correlated affinity with agonistic activity, and they found that the least affinity shows the highest agonistic activity. But that was published before, not before ours was published, because this is just accepted 2 weeks ago, but the studies were done 1.5 years ago. Hannah went for a 6-month world journey after her PhD defense. So okay. And IgGs that also bind, and now the question is of the binding epitope. There's a contact region domain 1 on CD40 that shows a high agonistic effect. Yeah? So if binding to this region shows a high agonistic effect. Okay, so now we have we have these therapeutic antibodies. They are also called Chilops-74, and they bind to an epitope in the N terminus in the contact region 1, which is the most distant one from the cell membrane. That maybe also helps for the clustering if it binds to more distance. And as mentioned, IgG2b shows the highest agonistic. So the most rigid antibody shows the highest agonistic activity. So now, and the questions that we raised is, do the differences in the flexibility of the different IgG subclasses have an effect on kinetics and mechanism of binding? Yeah? So we wanted to investigate the binding mechanism. And also, is the agonistic activity of CD40 ligand higher compared to the antibody? It is higher. And so the question is, what are the differences in the binding kinetics? Yeah, if we compare the CD40 ligand with the different classes of the antibody. And of course also, can we learn more how the clustering of the CD40 in the cell membrane occurs. So what we did is that we, let's start with the antibodies. So we bound all 4 antibodies to the AFM tip and studied the interaction with CD40s. And similar to the first talk, we were using living CHO cells in which CD40 was expressed. Okay, so experiments are done on cell. Okay, and this is what we found. So we found single and double bonds, sometimes also no interactions. Majority is single bonds, or also which can, that is similar as with the RBD. If we have double bond formation, they can dissociate sequentially or simultaneously. Okay, that is the same. And first of all, we found a little bit of a correlation in the sequential dissociation of the double bonds. That you can see that here, that the most flexible antibodies had more sequential double ruptures. Okay, so this is the stiffest one. I mean, it's not a huge difference, but there's some correlation between the number of sequential ruptures, which kind of correlates with the flexibility. So the more flexible the antibody, the more sequential these ruptures. Then we vary the contact time, and so first of all, with the RBD, you saw very nicely, 3 maxima, yeah, for single, double, and triple bond. Here, it is more hidden, and this made the data evaluation really intricate. Yeah, but we didn't come. We first tried to kind of tackle that with with one maximum, but it didn't work out. What you can see is that the shoulder becomes wider if we increase the contact time. Okay, so then we fitted 2 Gaussians in, and you can see that the number of the double bonds. So this is the overall experimental probability density functions, but they have showed us. And if we fit now 2 Gaussians in, we can see that the probability for double bond disrupture increases, and we can see that the number of double bonds increases with time. Yeah, so if we increase the contact time, so that means, as similar with the RBD, a second bond is formed, but in a timely manner. It needs some time to form the second bond. Okay. So then the first, what we did is we measured the K-on values. Yeah, you know that, binding probability with contact time, vary the contact time, and fitted the data. And what we can see is that the kinetic on rate is very similar for all 4. Okay, and this was also confirmed with surface plasmon resonance spectroscopy, which actually shows also that our K-on measurements are really very reliable. Yeah. Okay, so this was the first result, very similar K-on values. And then we also followed the time decay of the formation of the second bonds. Yeah, also what similar as we did it with the spike. And here we see some difference. Also both in SPR and in single molecule force spectroscopy, because it seems that you can see that here the values decrease from here to here, and this is the stiffest antibody. So this means that the stiffest antibody forms the second bond slower than the more flexible antibody. Okay, you can see the it. Hmm? Yeah, no, I agree that the difference is not great, but there is some correlation. Yeah. Pardon me?
technique would be better.
Yeah. I mean, as we get similar values, I would say for this it is fine. However, what we don't get, so we cannot with the SPR measurements, we could not get the K off values. They seem to be irreversibly bound, yeah, because there is some rebinding effects, and so we only fitted the K on curve. Yeah. So for all together, then AFM is better because there we could get the kinetic off rates, as you will see. How can we distinguish between those? I can't come to that because it's yeah. I mean, I come to that when, okay. And this is now exactly your question. I mean, I will explain it with the kinetic off rate, but there we also need it, yeah. How can we, so when we measure, we get this data cloud, okay? And this comes from single bonds and double bonds, okay? Again, we plot unbinding force against the loading rate and get this data cloud. So what we did was, and I mean, the first thing you may want to do, yeah, is to just take the data at different loading rates, yeah, fit two Gaussians in, and then separate it, yeah, and take, let's say, between plus minus sigma from the first Gaussian, the data, and put it back for the fit, and then take, yeah, and then and distinguish them and separate them this way. Yeah? But the problem is that the pulling velocity and loading rate is dependent on the elasticity of the system. Yeah? And this can vary a lot on the cell surface. Yeah? So we decided to do something else. We took this, we had to do some pinning, but we didn't want to do pulling velocity pinning, we did force loading rate pinning, okay? Because this decreases the error. So we beamed the data like this in different force loading rate, and then fed these points into an experimental density function. Yeah, so that's the experimental density function from this spin, that's from this spin, and so on. Okay, and then we fit it all with two Gaussians, and you can also see it's a little bit small, that this, the probability for double binding increases here. And why is this the case? Because with higher speed, the single and double bonds are better separated. Okay, because the forces become higher with loading rate, yeah? and with higher forces, the single and double bonds are better separated. Okay, and then first of all, we can fit now the most probable force, okay, and can say, okay, these are the forces that around this one. You see, you get nicely a Bell-Evans relationship, and around these forces around, and fitted a slope in there with the Bell-Evans model, and from this we can also deduce the kinetic off rate, okay. And this, and then we calculated the Williams model for multiple bond, and these are the forces for the double bonds. Okay, so that's how we did the data analysis. Okay, and you can see that here again a little bit better that for these are the bins from 10 to 32 piconewton per second and so on. But you can see that single and double bonds are better separated. And this is again, you know, kind of strengthened our thought that we indeed measure single and double bonds, okay? Because otherwise this shouldn't happen. Okay, and so what we can deduce is the kinetic off rates for that we measure, and we can see that the kinetic off rate is very similar for all four antibody classes, which is not so surprising because they all have the same paratope, yeah? And there's no reason why it should be different. And then we can also calculate the kinetic off rate for the two bonds. That is actually very simple. You can calculate it from the Williams model, and this is just a factor of 1.5. Yeah? So let's say the difference between an FAP fragment and an antibody for the dissociation of the whole molecule. Yeah? For 1 bound, it's the same as for the antibody and the FAP fragment. But then for 2 bound, the dissociation is just 1 times 5 slower for the antibody when you compare it with the FAP fragment. If you don't consider rebinding, this can happen, of course. Yeah? And this rebinding of the antibody maybe is also the effect why in Biocore the antibodies don't come off. Okay? Okay. So we would then say that the differences in the hinge flexibility do not critically influence the formation, a little bit the kinetic on rates, yeah, but not very strong, but not the kinetic off rates. So just rigidity, what we have indications, because we have lower number of sequential ruptures and a slight decrease in double bond formation. Yeah, this is what we have for the stiffer antibodies. Okay? But all 4 IgG subclasses can establish stable bivalent bonds. Okay? And the difference in agonistic activity has maybe more to do with the flexibility than on the cell membrane, more than with the binding. Okay? Because stiffer antibodies can hold 2 CD40 stronger together than more flexible antibody. Yeah? So we think this is more the reason for that, not really the kinetics of the binding. Okay. Okay. So then we looked at the CD40 with our high-speed AFM, and what we found is that the CD40 self-assembles on the mica surface. Yeah? So, I mean, this clustering is also physiologically important on cells, yeah, because it initiates these cascades, but it also associates on mica surfaces because they have an interaction domain. This is known. Yeah? So CD40 has interaction domain, and you can see that they form monomers. They also appear in monomers. I mean, you can see it really. We can nicely, and this is actually the crystal structure, and this is dimer and so on, monomer, and so we have monomers, dimers, and also tetramers, not so many trimers. Yeah? So it is more, they seem to associate more pairwise. Yeah? So they form dimers, they form tetramers, but not so many trimers. And then there's also some possibilities. So this is how it self-assembles without the CD40 ligand on a mica surface. So there are some inherent properties that they also tend to self-associate. Yeah.
How will you differentiate if there will be aggregates of these proteins from these?
Okay. So what you see here is also a probability density function of the volumes. Yeah. And so the same. It's also a purely experimental data. Here's and then we just fitted a multi-Gaussian function in there. Yeah. And this is how we can distinguish how many monomers, dimers, and trimers we have. Okay. So from the volume. Yeah, it can. There's also some. Let's say you can see that here. There's also some possibility for a pentamer. Yeah. At least with our resolution. But we see, I mean, the black curve is the experimental curve, and we see a clear maximum for monomer and dimer, for instance, and also for tetramer. Yeah? So they are definitely formed. Then, of course, it gets more difficult, but also this, I mean, here actually the black curve directly overlaps with the colored curves here. So there's also clear indications of heptamers. Yeah? And this is the probability density function of the volume. And again, I want to advertise the experimental probability density function. You never get this with a histogram. We look, so that, yeah, right, area and height, exactly, yeah. So we, let me just No, we fit them also. We fit them also, and calculate. No, we directly calculate the volume from.
Over a structure, the white height also might.
Yeah. Right. Yeah. So height and. Well, this goes. We put a mask on them, yeah, and then we have a program that directly calculates the volume. Yeah.
Height distribution there probably is integrated over.
No, it's right. You know, that is basically we take the image, yeah, and then we put a mask on, and then the program calculates the volume. Yeah. So because that is fairly easy for the program to calculate it. So we directly. I mean, of course, there's some tip artifacts or whatever. It will be a peer wider, but this is not maybe not so different for the pentamere or for them. That of course is something what we have to live with. We didn't correct for the tip radius, but for us it was only important to see the distribution and not. Okay. Then we recorded high-speed images of the, I mean, you saw the images there. But what we can see, I mean, I already said it, from the movies is that it seems, you can see that here. Here's two monomers form a dimer, yeah, so they associate. You can see that is taken from this movie, so we can observe it really also kinetically how it happens. Two monomers aggregate into a dimer, and here we have an octamer, which is hexamer plus a dimer. This is the octamer. So they kind of seem to associate mainly pairwise in a way. Okay, can self-assemble because it has an extracellular domain that induces self-assembly. And okay, it's more in an equilibrium between monomeric and dimeric state because this is what we mainly observe. And also, but tetramers and higher oligomers is mainly through coupling of dimers. And so this, what we see is it's already an inherent property of the CD40 that they can self-assemble, which is then important on cells. Okay. Then we imaged complexes between antibody and CD40. So what you can see here is the antibody. So this is the FC arm. This is a FAP arm. And you can see association of CD40. So here CD40 is bound, yeah, to the two FAP arms. Here one is bound. So we see association and dissociation. If it's associated, it's a purple curve. You can see it's kind of quite stably associated. So it can bind to both FAP arms. Okay? Yeah. I mean, you can see it in the picture kind of quite clearly. This is just the pure antibody, and this is the antibody with the two CD40s bound, and here also. Okay? I think you can see it quite clear, at least in the picture. Okay. So we have monovalent and bivalent interaction between IgG and two individual CD40 molecules. And the CD40 can also be in different oligomeric states, so it doesn't have, we see it with monomers, with dimers, and so on. Okay. So basically, it just confirms what we see with flow spectroscopy. Flow spectroscopy with CD40 in cells, and high-speed AFM is antibodies with isolated CD40, but in both cases we see binding of the CD40 to both FAP arms. Okay.
These high-speed AFM images, do we need to process it also, or these are the raw images?
These are the raw images, basically. And I can tell you it was a lot of work for Hannah to put these cycles on, because the problem with the first submission was that the reviewers didn't understand our figures. Yeah? And it was then even rejected, and we rebutted it and improved the figures. So in particular, these drawings, and then it was accepted without further comment. But what Hannah had to do is to put on a circle in each image, yeah? And with the sequence, and that's a lot of work. But these are raw data. Okay. Yeah, but what it seems, what helps here also, we seem to see a kind of rotation of the FAP arms, which then also helps to bind to the CD40. Yeah? So the antibodies are also dynamic, also a little bit similar as the spike protein. They are dynamic. Okay. So we see also with the high-speed AFM either monovalent or bivalent interaction between IgG and two individual CD40 molecules. And this is with CD monomers and also CD40 associates. And so basically, with all three methods, high-speed AFM, force spectroscopy, and BioCore, we see the same, yeah, that two FAP arms of the antibody can bind to the CD40. Okay, now we come to the CD40 ligand. So we put the CD40 ligand on the tip and measure interaction with CD40. And we also see only double bonds. Yeah? So this confirms this X-ray crystallography structures that also only see that 2 CD40 binds to CD40 ligand, although it's a trimer. And this is also what we see. We never see 3 peaks, yeah? Only 2 peaks. Yeah. Right. This is simultaneous rupture of rupture of 1 bond, simultaneous rupture of 2 bonds, and this is sequential rupture of 2 bonds. Yeah, never three. And experimentally, we can do it because with the spike we see 3 clearly. Spike is a trimer, we see 3. This is a trimer, we see only 2. Okay, so we did then the same game. Measured the kinetic on rate for single and for double bonds, and did this also with SPR. And here we can see that the kinetic on rate, both for the molecule and for the double bond formation, is clearly higher for CD40 ligand. If we compare it, so kind of rather similar, there's some correlation with flexibility of the antibody, but you can see kinetic on rate 1.6, so it's an order of magnitude higher, the kinetic on rate, if you compare it to the antibody. And also the kinetic on rate of bond formation, of the second bond formation, is much higher. Okay? Good. So the CD40 ligand seems to bind faster. Then we did the same game for the dissociation rates, and here we can see that the I thought I put this. No, but you can see it. Sorry that you can not see the letters, but you can see that the kinetic dissociation rate is also much faster, 10 times faster. Okay. But then, of course, the affinity is comparable. Yeah. So I think so. If we compare this CD40 ligand with the antibodies, kinetic on rate is 10 times faster, kinetic off rate is 10 times faster. Okay? And then we also looked at it with high-speed AFM, and here we also see that only 2 bonds are formed. Yeah? So the blue cycle is when it's bound, yeah? And we can see, so that we have 3 binding sites. They can bind to all 3 binding sites. CD40 can bind to all 3 binding sites, but only to 2 at the same time. Yeah? So the binding sites are equal, and also X-ray crystallography doesn't find any differences. 3 equal binding sites. CD40 can bind to all 3, but only to 2 at the same time, at maximum, either to 1 or 2. And also isolated CD40 and aggregated CD40. But then, if we observe, basically what you see here, yeah, see in this recording, if we observe that for a long time, 40 minutes, yeah, this is a recording after 40 minutes, and then you see a large associate of everything, okay? And this is something because that is, I mean, of course, it's done on the microsurface, but this is also what people see on a cell, that it leads to a large clustering of the CD40, and we can also see that with the isolated molecules on mica. Okay. Okay. Okay, so comparing CD40 with the ligand shows a similar affinity when we compare it to the antibody, but the kinetic on rate is 10 times faster, kinetic off rate is 10 times faster. Of course, if we compute that, then the affinity is similar. Maximum of 2 bonds with CD40, and the third one is always unoccupied. Binding is with CD40 monomers, but also with higher ones. And then cluster formation is driven over time. Large cluster formation is driven over time. Okay. Yeah, so these were the questions that we ask. There is a difference in kinetics. And what we think, why the CD40 ligand is so effective. So we can see this is the model we have, K-on, so that the kinetic rates for on and off are similar. So we think that the CD40 ligand acts as a kind of collector. Okay, so on the membrane, yeah, we have the CD40 in the membrane, and then the CD40 ligand comes. It can fast bind, yeah. During binding, association occurs, but then it can also fast dissociate. It can bind to the next one, and so on. Yeah. So basically, the collection of CD40 for aggregate formation is much faster for the CD40 ligand than for the antibody, because it associates faster, then it's aggregated, it dissociates, and can collect the next one. Okay, that is the idea. Because in fact, the CD40 ligand is of course a rather stiff molecule if you compare it to an antibody. Yeah? Antibody, the arms can rotate, but CD40 ligand is a kind of stiff molecule. So CD40 pre-assembly may also be important. So the self-tendency of CD40 already, but the driving factors for assembly are fast association and dissociation rates. Okay, and paper has just been accepted. Okay, thank you very much. Open for further discussions.
Peter, to show such dynamics or to prove that this is actually happening in the AFM, how many images you need to show, or how many videos of such kind you need to show, or is one enough?
Depends on the quality. No, one is not enough. You want to be sure that you observe the right thing, and you always also saw different videos.
Like these videos, one enough, or you?
No. Also different. You first have to figure out the incubation time and concentration and buffer and so on. Yeah. So but I mean, if you know the conditions, still you certainly record five videos. Yeah. But it takes some time to work out all the conditions.
So n equals to 5 is enough to prove that this is actually happening.
Yeah, if you see 5 times very similar effects, then I would be convinced.
With the third binding site.
Yeah, but for at least you need, in theory, just 2 sites, right, to bring things together. But certainly, I think it's also an advantage. So, on top of the higher association and dissociation rate, I guess it's also an advantage if you have 3 possible binding sites. Although you can only occupy 2 at the same time, but if you have 3 possible sites, it also would increase to the speed of cluster formation.
And for you, it is not possible to number them. That okay, if 1 or 2 is binding, then the third.
Yeah, of course. If you look at the videos, is here. So we have the 3 binding sites here, here, and here. Yeah, and we see that it can bind to all three binding sites. Yes? So we see it sometimes binding here, sometimes binding here, and sometimes binding here.
No, I was saying like, can you identify which, what is site 1, what is site 2, and what is site?
The three are equal.
Two are equal.
Yeah, it's a homotrimer. They are all equal. Yeah.
Okay.
I mean, so you can, but we could number them 1, 2, 3 here. Yeah. Because it's the same molecule, and we see it binds to all three, but not two at the same time. Not to three at the same time. Yeah.
So AFM basically images those molecules that are attached to the surface. But these molecules are kind of floating and then interacting with the molecule, the CD40. Then how is it happening? Or these are tracing onto the surface and then reaching to this CD40 molecule?
Yeah, but this is what you need to have. Otherwise, you cannot observe association and dissociation. Yeah, and this is the trick, to adsorb them to the surface, but not so tight that they are completely immobile. So just in a way that they can still move around the surface.
So this kind of rolling onto the surface.
It's not a rolling. I think it's just a lateral movement. It's similar. It's similar in a lipid membrane. You know, the lipids are diffusing due to thermal motion. Yeah, and here they are also diffusing due to thermal motion. Yeah.
So this is happening on a membrane,
No, it is on mica.
or is it floating then transiently attaching and
I don't
then?
think so. I don't think so. If they are floating, then you kick them away. So they are always bound to the surface, but not so tight that they are completely immobile. I mean, actually, that is, I think that's always the case. I mean, you have your degrees of freedom, also molecules in solution, and I think that's always the case. But if you look at it with a normal AFM, you just don't see it diffusing. Yeah? You just see something blurred. Yeah? That is the difference. But it's always kind of. I mean, of course, if we are using moderate zinc nickel concentrations for adsorbing them, to bridge them to mica. That is, mica is negatively charged. Most of the proteins are negatively charged, and we have made very good experience with nickel to kind of bind them to the surface, that they are stable enough for imaging, but still have some motional freedom that we can observe such things like association, dissociation.
So you do this after attaching your protein, and then doing this treatment, and then flowing the other protein which we need to see the association.
Exactly, yeah. You can see that here, so we are using nickel, and then I think here we incubated it at the same time, if the picture is right. I think here we incubated just both proteins at the same time. At least Hannah indicated it like this. Here it was premixed, and let's see how we did with the antibodies. Maybe we can also see it from the picture. Ah, yeah. Oh, it seems. So it seems that we, in both cases, we incubated the molecules already before we adsorbed them to mica, and then we just apply a solution for some time, 10 minutes or whatever. I don't have the exact now, but it's written in the paper. And then we wash it with the buffer, and then we do the imaging.